one of the dogmas in the brain is that once you make it during development, the nerve cells that form it are never replaced. So as you know, the brain is made out of these fantastic, beautiful cells called neurons. For Cajal, he was also an artist. This is actually a painting from Santiago Ramón y Cajal, who was a famous anatomist from Spain, and defined the neuron as an element in the brain, as the unit, the cellular unit that explains how the brain works. These structures were the most beautiful structures in all of biology. If you compare them to trees, for example, Purkinje cells and other cells in the brain, and he didn't think that there was any other structure in nature that was as beautiful as a nerve cell. And so one of the things that he noticed, and it became a big dogma, is that in the adult centers, the nerve paths are something fixed, ended, immutable. Everything may die, nothing may be regenerated. And this is actually mostly true, unfortunately, for neurodegenerative diseases, where some of these nerve cells die, and they have dire consequences on the health of organisms, including humans. So it is not surprising that people thought that it was nearly impossible to make one of these cells once it had already happened in development. First, you need the birth of the cells. And for that, you need the progenitors, and this is what we now refer to as neural stem cells. So there's a big field of study of neural stem cells, and my laboratory has been very involved in identifying these cells, and I'll tell you a little bit about that. Then you need another process that's really fascinating, and I hope to tell you I hope you will understand at the end of my lecture that this is really fundamental. It's called migration. The new neurons are not born where they end up being hooked up to circuits, but they are born very far away, and they need to migrate and through very, very complex territories to get to the final position. So it was not surprising that it was very difficult to think that this could happen in a fully formed brain with all this complexity. But the final and most challenging fact is that these cells need to get integrated into circuits. So once the circuits are made during development, these circuits are formed by neurons, and it is thought that there are stable elements. For example, some people think that memories are stored within these networks. Therefore, if you take some of these cells away and replace them with new ones, you would think that some of the memories would go away. Am I speaking at the right speed, or yeah? Okay. I'm very sorry, I really cannot do this in Russian. It's a double challenge because English is also not my native language, it's Spanish. So it's a double bridge here. We're using a barbaric language like English, and then with two much more powerful languages like Russian or Spanish. So we began, my interest in this problem actually began when I was a PhD student, working with a great man, Fernando Nottebohm, who actually was very interested in songbirds. And he had used songbirds throughout his life. When he was a little kid growing up in Argentina, he was able to identify different species of birds just by their song. And he became very interested in the mechanism of song production in birds. And he went on to discover a series of brain nuclei that are essential for song production. But one of the amazing things that he found that some of these song nuclei actually grew during the summer when the bird is in full song, and they shrink in the winter. And he thought that perhaps this growth of the nuclei during the summer might be associated with the addition of new nerve cells. Is that clear? So he began looking for them, and he found them together with the student Steve Goldman. So demonstrated for the first time very clearly, there had been other attempts, that actually new nerve cells could be produced in a fully adult vertebrate. So then the mechanism becomes very intriguing just because of all the questions that are raised before. Where are these cells born? How do they migrate from the site of birth to their final destination? How do they get integrated into these circuits? So that's when I came in. And we discovered that the stem cells were these curious cells that people knew as radial glia. We call them radial cells. This is the inside of the brain, the ventricles. As you know, the brains have two big cavities called ventricles that are filled with cerebrospinal fluid. And on the walls of these cavities, there are these cells with these long tails. And we found that these cells were the ones that were dividing to give rise to the new neurons. And this is actually an old technique that's not used very much anymore. It's autoradiography for the incorporation of truncated thymidine, showing that this cell has divided. So we correlated the division of these cells with the production of new nerve cells. And at the time, this was a big surprise because these cells were considered glia. And as you know, glial cells are considered totally separate from neurons. So what we found is that in the canary, there's actually, the epithelium is composed of two types of cells, ependymal cells, the classical adult cells that with these cilia move the cerebrospinal fluid, and these radia glia that in other species of mammals actually disappear by the end of life, and these cells divide to generate the new neurons. So actually this began changing the way we thought about who are the progenitors for adult production of nerve cells, and actually has now changed also the whole concept of progenitor cells, not only in adulthood but in development. I'll tell you a little bit in a few slides. So when I began my own lab, unfortunately, there was very little interest in songbirds. So we decided to move into mammals, and my first graduate student demonstrated an amazing process that happens also in the mammalian brain. Just like in canaries, the mammalian brain has large ventricles inside of them, and also just like in birds, in rodents, and this is the case of a brain of a mouse, there's very large numbers of progenitor cells there that actually generate neurons that go through this pathway into the olfactory bulb called the rostral migratory stream, and this was first described by Joseph Altman in the 60s. And Carlos Lois, this graduate student in my lab, was able to show that cells born here migrated very effectively along this pathway and became fully adult neurons in adult animals, became fully mature neurons that became integrated into the surface of the olfactory bulb. The olfactory bulb is called the olfactory bulb because it's the first relay where information from the nose, from the olfactory receptors coming, it gets processed here and then sent to the olfactory cortex. So it's a very, very essential place of the brain for processing of odors. So these neurons actually migrate along this path, and once they get into the olfactory bulb, they do not differentiate into one type of neuron, but they differentiate into at least 10 different types, and there's 6 of them illustrated here. They're actually migrating through the core of the olfactory bulb right here. Then they take off, and they stop migrating, and they develop these beautiful dendritic fields that are actually essential for the integration of olfactory information. So the olfactory information comes into this structure called glomeruli. There's also new neurons around glomeruli, and they get processed by big cells, mitral cells and puppet cells that are in these layers. And those cells are not forming at all, only these smaller cells called interneurons. And most of them are GABAergic. They are inhibitory. They produce the neurotransmitter GABA. So this actually set us up for life, and we've been studying this process for the last 25 years. And I'm going to tell you now a series of stories that have led us to understand how this works, the mechanism of this process. And then at the end, I'm going to tell you very recent experiments that show where these stem cells give rise to these new neurons come from. And that's going to be the main topic of today's talk. Unfortunately, there is no time to tell you, but there's many other interesting questions associated with these. One of them is, what is the function of these cells? Why does the olfactory bulb continuously integrate new neurons into their circuits? And if anyone is interested, we can discuss that after the lecture. But I want to tell you about the mechanism of making these cells. So most tissues, and this happens throughout biology, have a period of development, neurogenesis, that to me is one of the most fascinating problems in biology, when the tissues and organs are built. And here again, we allude this process to stem cells that through what people believe is a process of cell renewal, they produce progeny, they regenerate another cell that's similar to the original, and by this process they keep accumulating cells that become assembled into these beautiful structures that form organs and tissues, and in the case of the brain, form the nerve cells. So at some point this developmental process of constructing an organ stops, and then there's a maintenance process, and most tissues have stem cells that remain there to maintain the productions of cells throughout life. I mean, examples of well-identified tissues that keep producing cells throughout life is, for example, the hematopoietic system, the liver, the skin. But the brain was considered an exception. It was thought that it was constructed, but then stem cells stopped existing, and there was nothing of this sort in the adult brain. So as I alluded in the previous slides, actually the fact that we have found both in songbirds and in mammals a process of continual generation of neurons suggested that there must be some of these cells left in a fully mature brain. And this was a very interesting question, and many labs rushed into trying to figure this out. And I was very lucky to have in the lab a Canadian student from Montreal, from McGill University, that came to my lab and was actually, in collaboration with a group in Spain, able to identify these stem cells in the adult mammalian brain. Her name is Fiona Dodge. Now she's professor in the Biocentrum in Switzerland. And what she found is that the cells that generate the new neurons are these cells that are called B1 cells. And we call them B1 because they were the second cell that were identified in this area. We identified these small elongated cells called A cells, and I'll tell you what they are in a second. And then we identified a cell that looked like an amoeba, a more globular cell that we call C cells. So this second cell had been seen before us and had been diagnosed to be an astrocyte. Astrocyte is the most common cell in the brain. It's considered a glial cell, and it's called astrocyte because it has many processes. Some people call it star-like cells. And these cells are thought to be the support for the rest of the brain. They are the main glial cell for brain function. So nobody had suspected that a subtype of astrocyte could function as a stem cell. Now, as I showed you before, the cells that we identified years before in canary was also a glial cell, a radial glia. And there's a very close connection between radial glia and B1 cells, and I'll show you that in a second. So this began changing the concept that people had that neurons and glia are very separate, and suggested that at least some types of glia can work like as progenitors of neurons. So this was in the late 90s, early 2000. Then I had another great student, Saman Mirsadeh, actually an Iranian descendant that lived in California, and he actually made a very, very important discovery. He's by the, I don't know what happens to the screen, he's not that tan. It looks like he had been in the sun here. It looks different in this light, so the colors are a little off. But what he discovered is these beautiful structures inside the ventricles. So we were a little bit lazy, and instead of doing sections and histology, what he decided to do is just a whole month preparation, open up the ventricles and expose them. And then he stained them with a marker of the borders that stain the borders of the cells, this beta-catenin, and then a marker for the basal bodies of the cilia that move the cerebrospinal fluid. Well, he found these flower-like structures that we call pinwheels. Does everyone know what pinwheels mean? No. I don't know what's the name in Russian. How do you say it in Russian? In Spanish it's reiletes. You know little kids playing in the wing with these things that turn? How do you say it in Russian?
Флюгер.
Nugen.
Флюгер.
Я думаю, имеется в виду ветряная.
Baluga. Baluga?
But I guess everybody understands now, whatever the
Okay.
word
We
is.
got the idea.
Okay. So that's what a pinwheel is. That's what their name comes. And if you look carefully, the interest is not in these big guys, but the guys that are in the center. There's very small apical surfaces here in the center. You see here is a group of 1, 2, 3, and there might be smaller ones there. Well, these turned out to be the stem cells. So they're stained in blue there, and then if we use a marker typical for glial fibrillary acidic protein that stains these cells, actually it stains the centers of these cells. So this actually changed our concept. These actually were astrocytes that were touching the ventricle, making them more in common with radial glia. There were actually epithelial cells that had an apical contact with the ventricle. Well, more recently we had found what this apical contact might be doing. And this is not the whole story, but another great student, now someone from Singapore, Chupa Tong, came to the lab. This is Chupa. She also has finished now. She works at a biotech company in the Bay Area. And we find these structures inside the ventricle. These are axons. Axons running through the inside the ventricle. They're supraparenchymal axons, and they come from a very small group of neurons in the back of the brain called the raphe nuclei. I don't know if you have heard of these nuclei, but this is the source of serotonin. As you know, serotonin is very important in depression and other diseases. So it's surprising that there's a network of axons that runs on the surface of the ventricle and actually contacts the B1 cells through these synaptic-like structures. So these B1 cells, as stem cells, are working in adult brain, but they're fully integrated into the rest of the tissue. And this actually summarizes what we know about them. They have this apical contact, where they contact these, illustrated here, these long structures that are axons. They come from this small group of neurons that produce serotonin in the raphe. They have the cell body right embedded in the epithelium, and they actually have a long basal process that ends in blood vessels. So as you can see, there's multiple levels at which these cells can be regulated. and they look more and more like radial glia. They're epithelial cells, and they have long processes. They're not like radial glia that goes sometimes to the surface of the brain. Instead, they end in blood vessels in end feet. These end feet are actually typical structures of astrocytes. So now many labs around the world have been studying regulation of these cells, and actually we now know that they're regulated at multiple levels that integrate signals from the blood, from the serum, from the endothelium, the cells that cover the blood vessels. They're actually receiving signals from other progenitors within the structure, and I'll tell you what these cells are in a second. And they're also receiving signals at the apical bend, both from ependymal cells and also from things inside the ventricle and these axons that have serotonin. So they're integrating a lot of different signals from different places, probably to tell them when to divide, how many neurons to produce for the olfactory bulb. So they produce these new neurons not directly, but this is the lineage. These are the B1 cells. And actually, they produce neurons through an intermediate progenitor that happens to be this moamegoidal brown cell that we call C cells. These cells begin expressing proneural genes to make neurons, and they differentiate very quickly into these red cells that are the migrating young neurons. So these are already neurons. They're just very mature, and they're actively migrating from these sites where they are produced to go to the olfactory bulb. So just to remind you, these cells make at least these types of neurons. And to do that, they have to go a long way from the walls of the lateral ventricle into the olfactory bulb. This process of migration through adult brain is something that has attracted our attention for many, many years. And this is work from another graduate student, Hinek Viktorle, from actually a Czech, from Czech Republic. He's now a professor at Columbia University. He actually was able to make movies of these cells migrating, and we describe a new form of migration called chain migration. This is how these cells are moving through the walls of the ventricle, then that's the way they join this migratory stream, and then they leave the chains and they migrate individually, radially, into the different layers of the olfactory bulb. So these cells are migrating here, and they're making different types of neurons for the olfactory bulb. I showed you in the previous slide the different kinds of cells that they make. So the way we used to think about this, and we were totally wrong, and actually the way many laboratories around the world were thinking about this, is that the cells that were coming here were tabula rasa, did not have any defined property. And once they came into the olfactory bulb, depending on where they stopped, and depending on the local demands, they differentiated into different types of cells. In other words, they were multipotent once they were migrating, and they acquired these specific properties once they ended in different places. So let me tell you a little bit more details about these cells. These cells and these cells might look very similar to you, but they're actually very different. These are deep granular cells, and you see their dendritic tree arborizes deep in the external plexiform layer. These cells specialize in contacting the lateral dendrites of the big neurons that are here, the mitral cells. These are two types of superficial granular cells, and again, they might look very similar to you, to each other, but these cells, for some reason, express this protein called retinine, a calcium-binding protein, and these do not. And as you'll see, they have very different origins. And then there's three different subtypes of periglomerular cells, also identified by different markers. These cells express cal-binding. These cells express dopamine. They're pH positive, tyrosine hydroxylase positive. And these cells, like these ones, also express calretinine. So as I said, our idea was that the cells in the chains were multipotent, and then they were acquiring this property in different places. And here, another graduate student in the lab made a very important observation that began changing our mind. When she looked at the chains of migrating cells, there were heterogeneity among cells. There were, for example, some cells that their nuclei was clearly PAX6 positive, and this is a transcription factor that's very important to make those dopaminergic cells that I showed you before, and a subgroup of superficial granular cells. So we decided to say, maybe we've been thinking about this whole time wrong. So at this point, we really did not have the tools to really demonstrate where this heterogeneity was coming from. How would the cells that were already migrating the chains were made different? Another graduate student came to the lab, Florian Merkel, together with a postdoctoral student, Tony Tramonti, and developed a unique technique that has been particularly useful to a lot of experiments in the lab. What he did is something that we could not do genetically. He he came stereotaxically to different parts of the brain and micro-labeled the radial glia in different regions before these cells became adult stem cells. So what this showed is that the adult stem cells were actually linked to radial glia, right? I told you radial glia in birds were stem cells. Well, radial glia in mammals during development are also stem cells, which was a big surprise showed by multiple labs. Well, what he showed is that the adult stem cells were coming from radial glia, and he could micro-label them in different regions. So for what I'm going to show you now, it's not important that they are coming from radial glia, but the fact that he developed a method by which he could label them dorsally or ventrally, in the back or in the front. So in this way, he was able to make maps of the fate of radial glia in different locations in the developing brain. And what turned out was this beautiful concept. Different types of neurons in the adult brain come from different subregions in the developing, in the adult subventricular zone. Is that clear? So when he micro-labeled this domain, he got a lot of the Th positive cells and subpopulations of the superficial granular cells. When he labeled the ventral domain, he got the calbinding cells and the deep granular cells. And actually these cells that look very similar to this were not coming from the same wall, were coming from the opposite wall. Actually, the ventricle has two walls, one looking at the striatum, one looking at the septum. Here's the midline of the brain. When he labeled this side, then he got these calretinin cells that I was telling you, both the periglomerular and the granular cells. So with this method, he also went to label a region that in development is not known to make GABAergic interneurons, the inhibitory interneurons, the cortex. So the cortex, as you know, is the part of the brain that develops incredibly in humans and makes most of our brain. Well, it's now well established that the cortical interneurons, the GABAergic interneurons in the cortex, do not come from the cortex itself, but come actually from the ganglionic eminence. I'm not going to talk about that. So it was quite surprising to find that when he labeled the cortex, and see he's labeling groups of radial glia, many of them turn into astrocytes. This is in the adult, it was much earlier. So these are radial glia that go like this towards the surface. When you look at the olfactory bulb, it's full of neurons. So not only the lateral wall and the medial wall, but also the roof that gives rise to the cortex in development has the stem cells that give rise to specific subtypes of neurons that go to the olfactory bulb. And this happens in areas in front of the brain, all the way in the back of the brain. See, this is at the level of the hippocampus. Here's part of the hippocampus. And again, he's labeled the radial glia dorsally. So this was discovered by Florian Merkel and together independently by a laboratory a laboratory in Columbia University. This technique is actually so powerful that then he began actually poking and making labeling in different subregions of the brain. And we actually were surprised that when he goes very anteriorly and he labels the medial wall, looking at the septum, or the lateral wall, looking at the ventral basal ganglia, ventral striatum. When you look in the olfactory bulb, there were new subtypes of neurons that we had not seen before. So what this indicates is that heterogeneity is very great among stem cells, and that depending on their location, they give rise to different types of neurons. And there are some unique types of neurons that are actually derived from these very, very peculiar small domains. So I'm going to summarize with a little movie what I've been telling you up to now. I've been telling you that there is neurogenesis that happens on the walls of the lateral ventricles. I've been extending and saying that most of the walls, roof, lateral, and medial, that give rise to these new neurons, and that these locations seem to be already pre-specified to generate different types of neurons for the olfactory bulb. So if you were to make a section of the olfactory bulb, there are neurons, for example, from the ventral region that become superficial granular cells. There's cells that then migrate a little far. So this is the subgroup of neurons in the very anterior part, the four groups that I told you were just discovered. Then there's the periglomerular calbinding positive cells, the periglomerular cells from the dorsal region, the deep granular cells, superficial calretinin positive cells, superficial non-calretinin positive cells, and also the calbinding positive cells. So different subregions within these walls of the ventricle are giving rise to different types of neurons. So just to orient you, I know that many of you are not neurobiologists. This is the midline of the brain. There's two hemispheres. This is the cerebellum. Here would be the spinal cord. This is the forebrain. and this is the olfactory bulb. So are these ideas clear up to now? Yeah. So actually this might be a good point just to pause and take questions.
Yeah. So the suggestion is that you ask questions related to the first part of the talk now, so that it's easier to follow the second part, right?
Yeah. And if someone is not wanting to ask in Russian, they can ask in Spanish. If you don't want to ask in Spanish, you can ask in Russian, and then Anya will translate.
So, are these neurons only coming to olfactory bulbs? Or
Yes. In rodents, they are only going to the olfactory bulb as far as we have seen. There is evidence from a group in Italy that in rabbits, there is some of these cells migrating dorsally in this hollow part of the forebrain, but nobody has shown them integrating into circuits like it happens in the olfactory bulb. Our lab has shown that in humans, in children, very young stages in children, some of these cells go actually to premedial frontal cortex. And also go to the olfactory bulb, but this migration to the olfactory bulb in humans, in our laboratory, we have not found continuous throughout life. It's only happening in infants at young ages. Yes?
But what about migration to the stratum? I saw some books, I don't remember the name or the lab, but the books were about the migration to the stratum in humans.
Yes, there are several reports claiming that these cells can go to the striatum when you make a lesion. So when they occlude in medial irrigation, arterial irrigation into lateral striatum, they make big lesions, and some of these cells come out of here and go towards that area. Now, there's also evidence that lesions to cortex can lead to migration both to the striatum and to the corpus callosum and to the striatum and the cortex. Now, we have studied a little bit of this, and it's very controversial. And my impression is that these are not cells going to the striatum to make striatal cells. These are actually cells going to the olfactory bulb that get derailed because of the lesion and start migrating towards the striatum. If you look at the long-term survival of these neurons, it's very short. They don't live very long. And they don't seem to contribute to the repair of anything within the striatum that was the hope. We have also transplanted, taking cells directly from this area and putting them in the striatum or in the cortex. And they make cells that look like olfactory bulb neurons that do not live very long, and they don't seem to be well fitted for those regions. Yeah. And another thing is that when you make any lesion to the brain and you produce any edema, our impression is that this very small corridor becomes a traffic jam. And then cells begin to accumulate here, and that's why they might be coming off from the walls of the ventricle. But this is, again, this is the opinion of my laboratory. Other laboratories think that this is a way in which the subventricular zone is responding to the lesion, which I don't think is the case.
Could you please tell a little bit more about marking these tiny regions of ventricles? Using which tools we can do that?
Yes, that's a very good question. So the way we did it, so if this is the wall of the ventricle early on, the radial glia have a very long process that goes way out here. So what Florian did was began putting things out here in the process, and he found that adenoviruses are very good at infecting the process, and the adenovirus is carried into the cell. Now, adenoviruses, unlike retroviruses, don't become integrated to the nuclei, they become episomal. So if this cell divides several times, you lose the label. So what we did is we used an adenovirus carrying the 3 recombinase, and we did this in a mouse that had actually a flux reporter in radial glia. I don't know if this is clear. So this is a stop, and then so this is one of the mouses that we used. It had 2 3 recombinase sites, a stop codon here, and it had green fluorescent protein. So when the Cre got transported here, it recombined this site put the promoter directly in contact with the GFP and turn the GFP on in these cells. So now we had a permanent label for this radioglia, and we could do this here, we could do it in the medial wall, or we could do it in the roof like I showed you. And this gave us a permanent label for these cells.
And another question, you
Yeah.
worked with mice or with rabbits?
Mice. This was all done with mice. And the trick to do this is that in order to get these radioglia, they are in the embryo, but targeting these regions in the embryo is very difficult. So what Florian found is that these radioglia persist at birth in mouse. So he developed a stereotaxic apparatus for baby mice, where he put the baby mouse in a little mold made of plaster. He put them sideways, and then we came with very small pipettes and deposited a little bit of the adenovirus carrying Cree in these different locations. And that's how he was able to label break. As you can see, this technique is also very powerful because it not only allows you to label the cells, but it also allows you to flog specific genes. So we have used also this technique to remove genes within the radioglia and see what's the function of different genes for the development of different types of neurons. So there is no time to talk about that, but some of the work is published, and I'll be happy to show you some of that.
Thank you.
And what is the approach of looking then into these processes in humans? You mentioned that they were shown in children, some of the processes. How is it done at all?
Yeah, obviously you cannot do anything like this.
No,
so the human inferences all come from marker expression and from pathology. So we get children's brain from our pathology bank at UCSF, and we process them histologically look for markers of neurogenesis. And one of the markers that we use is a marker that's expressed by young neurons that's called double cortin. That's a microtubule-associated protein that's essential for neuronal migration. So this has been a very useful marker to see where these neurons are. We also, to look for proliferating cells, we use cell cycle-associated protein like Ki67 to see where there's proliferating. And this is how we have mapped the regions where there's still persistent migration of neurons into circuits in the postnatal brain in humans. This has also been controversial because there was a group here in your neighbors in Sweden, which we had big fights, and a group in New Zealand claiming that in humans there was big-time neurogenesis going to the olfactory bulb. And they published this as a cover paper in Science. And this paper, we believe, is totally wrong. We have looked in with different conditions at what they claim is an open ventricle and a connection between the subventricular zone in humans and the olfactory bulb, and we just don't see it in adults, but we see it in infants. Up to like 8 months of age, it's quite strong in humans. But then it goes away. And it could be because olfaction is not that important for humans, and the other reason we have published recently is that because the human brain might grow so much, it might separate so much all of these regions that might make this migration just impossible. Yes.
What signals are able to make neural stem cells to proliferate and
Differentiate.
Yes.
So there is papers and papers about this, and we have done some of the work to do this. It's not totally well defined, but for example, some of the signals involved is FGF, fibroblast growth factor. EGF is also involved. EGF induces proliferation of these cells very strongly. BDNF has been claimed to be involved in the proliferation of these cells. There is evidence that vascular-derived growth factor from the vasculature is also related to the inhibition of TNF, has also been shown to be involved in making the cells proliferate. And there's both signals that make the cells divide, and there's signals that make the cells divide to generate C cells that end up differentiating and giving rise to the new neurons. There's also a great deal of interest in this system as a possible source of brain tumors. And here, with a group in Texas, we showed that actually the B cells can be labeled and can give rise to glioblastoma. But my impression, and I think now this group has direct evidence, that not the stem cell itself that one is responsible, but the C cell, that there's a failure in the break to stop them dividing, and they just keep dividing and accumulating mutations. And if you give some of these factors I was telling you, like EGF, you make these cells keep growing, and they actually start invading the tissue just like a tumor does. So there's also a great deal of these cells, a great deal of interest of these cells in terms of their possible source of tumors. Now, these cells, in addition to neurons, they also make oligodendrocytes. Oligodendrocytes are the main myelinating cells in the brain, and they are produced constantly throughout life, and they're produced from two sources. One is from local oligodendrocyte progenitors, so-called OPCs, and also from the subventricular zone that's able to replenish some of the OPCs, especially for the corpus callosum that's right up here, right in this region, it's right above here. So if you see it, some of the sections that I showed you before. So this is corpus callosum. And so these progenitors make oligos that migrate and populate this area. And if you make a lesion to kill oligodendrocytes in the corpus callosum, the production of oligos from the subventricular zone gets activated and they make more oligos for that region. So these cells can make at least two types of cells, neuron for the olfactory bulb and oligodendrocytes in the adult. Yes?
I just cannot resist asking you about the stem cells that lie beneath the ventricular zone. There are several diseases that usually affect this periventricular zone, like multiple sclerosis. And well, in other neurodegenerative diseases, we also see the enlargement of the ventricles. Are there any research in this area? Do these disorders somehow affect the stem cells of the brain? So as we can see for example, in multiple sclerosis, that after the stage of inflammation, you can see the degeneration stage, so which we cannot control. So probably, well, are there any research in this area if these stem cells somehow become damaged?
So as I said, in humans, it's not clear whether there is a lot of these stem cells left there. In mouse, there's models of hydrocephalus that result in ventricular increasing. And some of those are actually associated to problems with the animal cells, the ones that are right next that form those pinwheels. If there is defects on the movement of the cilia of the cells, on the polarization, it's another fascinating phenomena, how the ependymal cells develop. And actually in the lab we have studied some of the factors that influence ependymal cell development because they're very, very tightly associated with the B1 cells. But your question, I don't think, has really been addressed the way you want to address it, because what you're really asking is, in the case of hydrocephalus or inflammation, or are the B1 cells affected? And I don't think anyone has directly addressed that. What is known is that if you mimic a VMS lesion by killing some of the oligodendrocytes, as I said before, some of these cells then begin making oligodendrocytes that move actually into the site of lesion. But how hydrocephalus affect the niche where these cells are, that's very interesting. I don't think has been addressed. So much for all these interesting questions. Now I'll try to continue, and by now you should have know more or less about the process of adult neurogenesis. Actually, someone just asked me a very, very important question: are there stem cells anywhere else in the adult brain? And there is another region of the brain that's very, very actively studied where there's production of neurons, and it's the dentate gyrus of the hippocampus. And there too is a glial-like cell that we call radial astrocyte that actually is producing the new neurons. So now I would like to go to also questions that arise during the break. What is the origin of these cells and what is their behavior? So we know now that radial glia actually are also the stem cells during development. I could not show you the evidence from that, but it's not only my laboratory, but the laboratory of Arnold Krickstein, who is also at UC San-Francisco, demonstrated that radial glia divide and they generate neurons during development. They're actually the progenitors of the cortical neurons, the pyramidal cells. So the idea is that the brain begins with an neuroepithelium, and what is the connection to the adult stem cells? So we have all assumed that these cells go through the production of all the types of brain cells that happens to be produced during development, and then some of those cells turn into B1 cells, and those maintain the generation of neurons throughout life in the walls of the ventricle. But even though this was published in some reviews that we wrote together with Arnold Krickstein and by myself and with other people in the lab, we actually did not know this very crucial connection between the embryonic stages and the adult stages. And that's what I want to tell you about in the second part of my talk. So the idea, and based on the way we think that stem cells behave, and those of you that are stem cell scientists might know that one very important principle of stem cell research is what's called cell renewal. The idea that a cell can divide and generate another progenitor that's very similar to the original progenitor. And that's the way, for example, the hematopoietic system keeps production of enormous numbers of cells throughout life. So the idea was that through a process that approximated cell renewal, the cells went through a process of neurogenesis and gliogenesis during development. And then some of these cells became adult B1 cells, begin producing preglomerular cells and granular cells throughout life. Now, this is a very, very brief moment. I mean, development in mammals is very short. This can be very protracted. For example, in a human, development can happen maybe within a year. Then life can be up to 100 years or even more in some individuals. So the protracted nature of this process raised the question, what is the process that maintains cells for such long periods of time being able to generate neurons for life? So this was the concept based on stem cell biology textbook, but the truth was that we did not know at all what was this connection. So in the last part of my talk, I'm going to focus mostly in very recent work from a very, very talented postdoc. This guy now comes from another part of the world, from Chile. His name is Luis Fuente Alba. He's no longer in the lab, unfortunately. He has also moved to work for biotech in the Bay Area. And actually, he did an experiment that surprised me and I think the whole field. He did classical birth dating experiments. I told you about thymidine as a marker of division before. We used it a long time ago to label radioglia. Well, what now many people use is another analog of thymidine. It's called bromodeoxyuridine. Does everyone know what bromodeoxyuridine is? It's very widely used in biology to label dividing cells and get incorporated into DNA as the cell divides during the S phase. So what he did is he gave bromodeoxyuridine during development of a pregnant mouse. Then he let the pups develop till they were postnatal day 21. And he looked at the wall of the lateral ventricle and used the pinwheel technique to identify the B1 cells and found, to our surprise, that the majority of B1 cells were labeled by injections at E14.5 or E16.5, 17.5. So this was surprising for the following reason. You would imagine that if this model was correct, that if you were injecting bromodeoxyuridine here, by the time these cells are adults, they would have diluted down the BRDU and you would never get a peak. You see how that was surprising? So this began suggesting that this model was not correct. But still, we could not know if, for example, these cells would have diluted down by many divisions. So he did another experiment that does not suffer from the dilution effect. That's injecting a retrovirus into the lateral ventricle of the pups inside the mom. So you take out the mom, you do a laparotomy, you take out individual fetuses, and with a small needle you go inside the ventricles and you inject a small amounts of a retrovirus that carries the green fluorescent protein. Now retroviruses have this unique property. They get incorporated into the cell when the nuclear envelope breaks, so they serve to mark cell division also. So we found the same thing. The majority of cells that we found in the adult label were coming from cells that were divided in mid-fetal development around embryonic 15.5. So very similar results with two different techniques. Now, with this technique, we were able to look not only at the cells in the walls of the lateral ventricle, but at the actual neurons within the olfactory bulb, because we knew that this method did not dilute the virus. So the neurons in the olfactory bulb are beautifully labeled by a single injection of retrovirus at any one of these stages. Is that clear? Okay, so this began changing the way we're thinking about cells, suggesting that the cells that became the adult B1 cells were highly produced in a very specific period of development, suggesting that they were not like leftovers from development, but they were maybe set apart from division of progenitors at very specific stages of development. So if BRDU did not dilute down sufficiently, perhaps now we could use BRDU to label neurons all the way to the olfactory bulb. So again, we can give BRDU at that time when many cells are being born at this stage. And we do all of this in a mouse that carries a flux reporter specifically for neurons, so that if we give tamoxifen in postnatal stages, now we can label a generation of cells that go to the olfactory bulb. The tamoxifen through the flux gene would result in red cells, These are all the red cells that were produced from these tamoxifen injections. And the BRDU, we label it with green, so we can have a double label cell that was born in the embryo and was actually activated in the adult life. Is that clear? So now we can do two different experiments. We give the BRDU both at these stages, but now we wait either till day 15 or day 45. And the idea of waiting this length of time is that if cells were really continually dividing in vivo, by day 45 they should be much more diluted than by day 15. So when we compare the amount of labeling within the nuclei of the cells labeled at P15 or P45 is totally equivalent. There is no significant difference, suggesting that the cohort of neurons that got activated by the cohort of stem cells that got activated by P45 is different than the cohort of stem cells that got activated by P15, or that the level of dilution that happens between these two stages is similar. In any case, it goes totally against that model where the cell is constantly self-renewing. And this is causing a very big stir in the stem cell field, because people had assumed all along that actually stem cells were going to be self-renewal. So for the second part of my talk, the first conclusion is that after their generation in the embryo, around embryonic day 15 and a half, postnatal neural stem cells remain largely quiescent, and then they get activated at different times in postnatal life. It's almost like the embryo is preparing these sets of progenitors and setting them apart during mid-fetal development, because they are going to be the progenitors for adult neurogenesis. Now, what we really want to do here is map individual cells. That's really the most attractive method to really understand what is going on with these cells. Now, when you do clonal analysis in a system where migration is very tight, you can all make inferences about the cells that are close by and close together as being all clonally related and originating from one cell, and that's how people usually do clonal analysis. The other way to do it is to label individual cells and make movies of those cells and see what they become. None of those techniques we can really use here, right? There's a very long migration from the site of origin all the way to the olfactory bulb, and there is no way that we can put a camera in there to watch what the individual progenitors are doing. So what we decided to do is team up with a group in Harvard University in Boston, who actually has developed a technique that is now usually referred to as barcoding. And the idea is to put an individual DNA sequence within only one progenitor or a few progenitors. And the way they do that is generate a library that contains very high level of complexity, where this oligo varies in each one of the viruses. So there are 24 bases here, and there's 10 to the 7 theoretical combinations of these bases. Actually, when Konichevko's lab in Harvard measured the actual complexity, is 10 to the 5. So the likelihood that you inject this mixture of viruses into the embryo, the likelihood of two cells getting the same virus with the same sequence is extremely low. Is that idea clear? So using this tool, now we do the same experiment we did before. We do a single injection at different ages during development, but now we don't inject this single retrovirus carrying GFP. We actually inject this library, complex library, hoping that each one of these cells would get only one virus with one of these tags. One additional detail, this library was modified to include a membrane-bound GFP. So this is this is a green fluorescent protein, and it's tagged to the membrane. It has a little sequence that makes it go to the membrane. And what this allows us to see beautifully the morphology of individually labeled cells. So we wait for these animals to be born. Then we give tamoxifen to label an adult generation of neurons, and we do all of this in nesting Cree-ER mouse that were produced by this laboratory at UT Southwestern in Texas. And these animals contain a flux reporter in cells that are derived from stem cells that express nesting. So when we give tamoxifen, we label a generation of cells going to the olfactory bulb. It's too bright, the lights here, but you can see the olfactory bulbs of these animals full of labeled neurons just from these 4 injections of tamoxifen. Do you see that? So now we look for double labeled cells, cells that contain the GFP and cells that were produced in the adult. Now, the way we do these experiments, and they took actually 7 years to complete, was we take the whole brain and we section it serially from front to back. Wherever we find a GFP labeled cell, we microdissect it with special machine, a laser microdissector, take out that cell, actually amplify it by PCR, and send it for sequencing to try to detect what sequence was within that cell. Obviously, we cannot see those 24 base pairs just by looking at the cell. We need to send it to be sequenced. So actually, this works actually beautifully. It's difficult to do, but it's very beautiful because you can see here a double labeled cell labeled by the virus with GFP and labeled by the TD tomato. It was produced in the adult when we gave the tamoxifen after P30. This is a periglomerular cell, one of these cells, and this is a granular cell with this cell body here. So these cells were laser microdissected, PCR amplified, and sent to be sequenced. Now, when we looked at the cells that had the same sequence and we defined clones by having the same sequence, the first thing that was surprising is that actually the clones are very dispersed from front to back. This is the olfactory bulb, this is the front of the olfactory bulb, this is the back. This yellow clone, they all had the same 24 base pairs is this clone. They were found throughout the olfactory bulb rostral caudally. There's another clone that had 3 cells. They were also in different locations rostral caudally. Now, when you look transversely and plot the position of each one of these cells with respect to the core, their position deep to the surface, they were very clustered. Even though this clone was very dispersed rostral caudally, you can see most of these cells are deep granular cells. This other clone is superficial granular cells. And this began to make sense based on what I told you before, that the progenitors are very specified regionally. So when we looked at most of the clones that we found, were specific, even at various stages, early stages of development. When you injected the retrovirus at embryonic day E11.5, they were restricted to 1 layer of the olfactory bulb. So what this means is that this regional restriction I've been telling you about that happens because of the location of these stem cells actually is determined very early on in development. There were actually a few clones. For example, let me show you these ones. These are periglomerular cells. This was one clone, only contained periglomerular cells, no granular cells. This was another clone that contained intermediate and superficial granular cells, but no deep or no periglomerular cells. So isn't it surprising that you're labeling individual progenitors in the embryo and they already know what they're going to become in the adult based most likely on their location? I'm going to show you that direct demonstration of that in a minute. Now, there were a few mixed clones that we found, and these were very interesting also. And this is an example of one of them, and there were a few of them, but they were always of this type. They were actually superficial granular cells combined with periglomerular cells. And remember I told you about the PAX6 positive cells that could become dopaminergic cells and superficial granular cells? Well, these are them. So even those that were combined were very specific combinations of cells that actually come from the dorsal subventricular zone. So the second important conclusion from the second part of my talk is that postnatal neural stem cells become regionally specified very early in development. So this again is a steering up the stem cell field, because for many the fact that there were stem cells in the adult brain meant that we had very flexible cells that were going to be able to fix everything, you know, Alzheimer's, Parkinson's. There were these cells that were very at the root of lineages that were able to differentiate into anything. The way the brain is made suggests that these cells become regionally restricted very early in development. So in other words, I showed you in the first part of the talk that different regions gave rise to different neurons. So what this began suggesting that these different subregions were already specified in the very early embryonic stages. And whatever labels and molecular restrictions and epigenetic restrictions these cells had, they're already stamped by these early stages in development. So you can imagine that we not only had cells labeled in the olfactory bulb, but with this we actually had cells labeled throughout the brain. And this gave rise to a very, very interesting basic question that we could ask. Are cells in the olfactory bulb that are produced in the adult at all related to other neurons in the rest of the forebrain? So if we section serially the brain, we had examples of all kinds of neurons. For example, medium spiny neurons in the striatum, cortical cells, interneurons, and pyramidal cells. We had pyramidal cells mixed with astrocytes. These are two astrocytes that also occur in the same radial column as the neuron. And there were actually some cells left in the subventricular zone, which we're now studying, which are also very interesting because these might be cases of cell renewal. And we're studying if these cells are related to cells in the olfactory bulb. So now we can ask the question, are these cells that are labeled in the rest of the forebrain, has any of them had the same 24 base pairs, the same barcode that the cells in the olfactory bulb, perhaps indicating that they belong to the same clone? And the answer is yes, but this only happens in the very early stages of development. When it's adult, we do not see this happening. So this is actually looking at the cells that are born after tamoxifen and the cells that are born before tamoxifen. After tamoxifen, we found one clone that was lineage related at E11.5 and E12.5. Obviously, it's very few clones because the olfactory bulb has very few clones produced at this stage that we could map up to the E15.5. But we could also look at all of the cells that were not red, that were produced before the tamoxifen, and this is them. And again, here the indication is that early on, the cells have a relationship, but later on this relationship is lost. We can actually now move the tamoxifen injection earlier at P6 to now label cells that were born after P6, and now we find more cells, but again early, but not late. By E15.5 of 38 clones we found in the olfactory bulb, none of the neurons in that clone were related to neurons in the rest of the forebrain. So the third conclusion from this afternoon talk is, that postnatal and fetal forebrain neural stem cells share a common progenitor in the early embryo, but these two lineages diverge between postnatal day 13.5 and 15.5. So the last part I want to show you actually relates to the question, where are these cells in the forebrain that were related to those cells that we found in the olfactory bulb? And this actually links us back to the original specification. So this is a clone that actually had one periglomerular cell and several superficial granular cells. These are one of those mixed clones I was telling you. And we knew that these clones come from the superficial granular cell looking at the cortex. And where did we find the lineage-related cells? A little clone of cells that were radially oriented in the cortex. Now I'm going to show you this one very different. This is a deep granular cell. We know that this comes from the ventral side of the wall. And this clone was in fact related to a clone in the striatum. And the third one I'm going to show you is a superficial granular cell that is calretinin positive. Remember, these cells came from the opposite wall, the ones looking at the septum. And in fact, the clone that was related to this cell was in the septum. What this indicates is that the location is key. And location is the first thing that determines not only this clone was going to give rise to a few septal cells, but later on was going to give rise to a progenitor in the adult that was going to give specifically rise to superficial calretinin positive cells. So the conclusions from this later study is that after their generation in the embryo around 15, postnatal neurostem cells are not these self-renewing cells the way we thought, but are largely quiescent and they get activated at different times. The consequence of this conclusion, as you can begin thinking, is that neurogenesis in the adulthood is a finite event. If you're consuming cells as you're using them, the number of progenitors that you're left with are less and less with time, and this is actually what we're studying now, that with aging, the number of stem cells decreases tremendously. The second one, postnatal neural stem cells become regionally specified very early in development, suggesting that acquiring a location address is the first thing that happens. And then postnatal and fetal forebrain neural stem cells share a common progenitor in early embryo, but these two lineages diverge during the mid-fetal development. And perhaps the best ways to illustrate this is by bringing back the diagram that I showed you before and kind of implying that linear tree that we had, sort of thinking that the adult development was like a pine with a central trunk and the final branches at the tip of the tree were those related to B1 is really wrong. It looks much more like an oak with many branches from the bottom, and one of those early branches is the one that gives rise to adult progenitors. So there's very specific progenitors that diverge here that become the adult progenitors. And that populates this region that we call the ventricular subventricular zone. And these cells seem to be very unique and very highly specialized for the production of very specific types of neurons. So what I showed you today is that there's multiple cells that can be produced in a part of the brain that process olfaction. All of these neurons are GABAergic interneurons. They're actually involved in breaching and making little breaches between the projection neurons. So to some, this might be the minutia, maybe thinking that the big cells, the pyramidal-like cells, the mitral and tufted cells are the big deal. But I think to those, I would say that they're totally wrong. These are actually the key to brain function, because these are actually the cells that are regulating how these big cells are putting out information to the rest of the brain. And the reason why the olfactory bulb is keeping this process throughout life is not to repair anything, because the olfactory bulb is not suffering from any kind of damage. This is a process of constant plasticity. What I would suggest to you is that these cells are produced within this part of the brain to keep young neurons with some specific properties coming on throughout life, bringing on new circuit combinations to the olfactory bulb. And we can discuss why you need to do that with the olfactory system and not with other sensory systems. That's one of the interesting questions that follows up from all of these. But the fact is that these cells are very highly specified, and that the whole brain, the whole subventricular zone, is making different types of cells for this diversity of cells going to the olfactory bulb. So I think one of the challenges for the future is, can we induce similar processes in other parts of the brain that don't get normally new neurons? And I don't think it's going to be fixing the big wires. It's going to be fixing these local interneurons that's going to be possible. So my other half of the lab actually works in replacement of interneurons outside of the olfactory bulb. And actually there is no time to tell you about that. But just in combination to try to give you the general conclusion of what I've been telling you today. Location is key to development. And we know from studies from Tom Jessel, from John Rubinstein, and from many others that actually early in development, different transcription factors get segregated in different subregions of the wall of the ventricle. So I think these cells are inherent, and positional information is going to be key, not only for the cells that they're going to produce as embryonic stem cells, but for the cells that are going to be produced later on in life from progenitors that are derived from these cells at these stages. So what I would say is that these early domains in the early embryo get translated in the adult into domains that remain that way, but producing very different cell types. Again, in the mouse, all of this seems to be servicing the olfactory bulb. So one region that is very, very interesting in this context is the ventral forebrain. This is called the medial ganglionic eminence. And years ago, John Rubinstein's lab and then our lab showed that it's actually the source of interneurons that go to the cortex. So as these cells are making cells that go to the olfactory bulb, early in development, the MGE is a little factory that's making the interneurons that are going to form this network of interneurons for the cerebral cortex. About 20% of the cells in the cerebral cortex are interneurons. This is actually what Ramon y Cajal called the butterflies of the brain. And actually he thought that in humans they were more diverse than other species, and perhaps were the key to the higher intelligence of humans. So there's no time to tell you today, but the other half of my lab works in this question. Precursors of GABAergic interneurons from the embryonic medial ganglionic eminence, when transplanted into the postnatal cerebral cortex, migrate and integrate into functional circuits and can induce a new period of juvenile plasticity. Just like happens in the olfactory bulb throughout life, it can be induced by the transplantation of these other types of interneurons to the olfactory bulb. So with that as a future perspective for brain repair, I'd like to end my lecture today and just again thank the great students that I focused mostly on the subventricular zone today, and actually most of the second part of the work was the work of Luis Fuentealba, together with Kirsten Overnier. There was no time to talk about her work, but she's actually looking at the actual question of cell renewal in the adult brain. And this is actually a view of our institute, the Stem Cell Institute in San Francisco. This is the Pacific Ocean, and this is the west part of San Francisco. Thank you so much for your attention. Yes.
I don't know about hippocampus stem area. There are two radically different opinions about the ability of hippocampus neuron stem cells to be self-renewed. And what do you know about some things like that about ventricle stem area? Are there only one opinion that they are self-renewal, or
Yeah, so in the hippocampus, the question refers to about 2 years ago, Grisha's lab in Cold Spring Harbor actually suggested that there was a cell consumption of the radial astrocytes in the hippocampus, so that there was a finite number of neurons that were produced from that cell, and the cell renewal was limited. It's not known in the subventricular zone how it happens, but some of the data that I presented to you today is more consistent with the idea that it's finite also. Otherwise, you would find that the dilution of BRDU from the initial injection would be higher in a later generation than in a younger generation of neurons. But this is still very controversial, and in the hippocampus, as you were alluding, there is now other papers that have come out where they show some level of cell renewal with symmetrical division of radial astrocytes within the hippocampus, what all people refer as type 1 progenitors. Yes?
During your lecture, you told that it should be, the mechanism which you described should be the mechanism of neuronal plasticity, because in olfactory bulb, cells are not damaged during the life. Do you check it, or it's just your opinion? Because the nose and the olfactory system is a system which connects to the environment, and I have read the articles about that maybe nanoparticles of the air could came to the brain, and it's possible that this zone can be damaged. Do you know articles, maybe some papers about this fact?
Yes, yes. So there's many speculations. There's no direct evidence, but what you're saying is perfectly correct. But there is just one inconsistency. The olfactory epithelium is fully exposed to the air. It comes to the olfactory bulb here through the nerves, and this is already behind the blood-brain barrier. So the particles that are exposed here cannot get deep in here. The new neurons are being produced. The second evidence is that you can induce damage here, and if what you were saying was correct, that should increase the proportion of cells replaced here, and that doesn't happen. And the third evidence that what you're saying probably does not happen is that you can directly induce a damage in the olfactory bulb in one region. If these cells were replacing cells within the damage, you would think that there was increased replacement in the region of the damage. And in fact, the cells that are coming in do not go as frequently in the area of the damage. They prefer to go into the area that's not damaged.
Yeah, but I just ask if you maybe check for cell death in these areas. You make a lot of slides in different ages of mice. Can you check some markers of cell death?
Yes.
Yeah?
Yes. So
That you don't find.
no, there is cell death, and this is, as I was referring to, this is a mechanism of constant neuronal replacement. So the neurons are dying and are being replaced by the new ones, and actually the process of cell death is essential for the new neurons to come in. You have to create a vacancy for the new neuron to have a space to come in a new cell. So if you prevent the formation of new neurons to go to the olfactory bulb, cell death keeps going on in the olfactory bulb, and you get an atrophy of the olfactory bulb. But the process of cell death is not due to damage.
And you check it.
It's a process of programmed cell death that is part of the plasticity. You have to create new vacancies, remove all neurons to have a new one come in, and that's exactly what happened in the zone control nuclei in canaries. These are very, very good questions, and my impression that's part of the process of plasticity. So a very interesting experiment nobody has done is, can you prevent the cell death in which case you would prevent the formation of vacancies, in which case you would decrease the replacement of neurons. My impression is that what would happen is that the level of plasticity would go down. Now there's another whole area where the concept that I told you comes from, and that comes from bioinformatics. With a group at Rockefeller University, we created a parallel neural network that mimics the olfactory bulb. And the only rule added to this is that when you were adding new neurons, if they were active, they would survive. If they were not active, they would die. And when you do that, when you incorporate new neurons at random, cross-inhibiting the different lines of information coming through the olfactory bulb, you greatly increase the contrast of olfactory perception. And that's where the idea of plasticity comes from. There was not enough time to explain it today, but if you're interested, I can show you some of the data. I have it right here in the computer. Very good question, yeah.
Which triggers leads to the cell death?
That's another fantastic question that is not known. In Canaries, a little bit more is known because the difference in the time of the year are due to difference in levels of testosterone. Canaries' testes are incredible things. They grow 400 times between so some of the birds that you have surviving these winters here, I don't know how they do it, have very tiny little testes. By the spring, when they begin singing like crazy, their testes are growing like crazy, and they're pumping out testosterone. And what seems to happen in canaries is that the testosterone triggers the secretion of BDNF locally in the brain, and that actually stabilizes the neurons that are already there, new ones and old ones. In the fall, the levels of testosterone drop abruptly, and actually that destabilize the neurons that are there, and there's an increase in cell death and increase in the formation of vacancies. This is another idea that this is not related to damage, but it's related to hormonal changes to trigger opening of another critical period for new song components to be incorporated for the next year. But in mammals, nothing is known. That's nothing is known in the olfactory bulb.
Any other question? So everything is pre-programmed, and the neural stem cells, they are like perfectly timed, yes? And are you searching for some factors that can induce neuroplasticity?
Well, I wouldn't say that everything is programmed. That would be an exaggeration what I said. What is programmed is the types of neurons that these cells are going to make. There are areas where
they are, well, at certain points at time, how the time is defined, by what factors, growth factors?
Yeah. You're referring to what you were asking me before, right? The timing of induction of plasticity?
Yeah.
Yeah. So the question before was a very interesting one. When we transplant cells, I told you about the other part of my lab that is transplanting medial ganglionic eminence. So different parts of cortex that have different forms of plasticity. And one of the best forms of plasticity that have been studied is visual plasticity. When you close one eye, the connections in the cortex for the two eyes get adjusted only during a critical period of plasticity that happens around postnatal day 27 in mice. So what we now know is that this plasticity is largely due to the maturation of neurons that were born here, that at day 27 they induce plasticity. Now these are the MG neurons, medial ganglionic eminence neurons. We take these embryonic cells now and we transplant it into postnatal animals or even into adult animals, and now they're able to induce a new period of plasticity exactly when they are 33 to 35 days. That is equivalent to when the animal is P27, because there's a few days before birth when these cells are maturing. Is that clear? So from this came the concept that the timing of maturation of interneurons is key to the induction of plasticity. So in the olfactory bulb, what's happening is that you have these neurons coming in all the time, so you have an open period of plasticity throughout life. This is all conceptual and is based on other experiments that I didn't show you today. So what you're now asking me is what determines this period that the cell is going to induce plasticity exactly when it's 33 to 35 days. I don't know. If I knew that, there's something within the cells. There's some clock that is inducing plasticity there. There's a recent paper from Takao Hench where he has not transplanted, but he has actually looked at the induction of plasticity in clock mutants. You know that there is these mutants that determine the circadian rhythms, and these animals never go into critical period plasticity. So perhaps these cells are counting days, and it's just not known how it happens. But you know, all cells in the body, not only the arcuate nuclei and the cells involved in circadian rhythms, are keeping track of time, and perhaps they are counting days, and within that day something happens. The other thing that is known is that if you do genomics, proteomics analysis of these cells at different stages, or genomic analysis of genes that are expressed at different, there's very dynamic, and there's all kinds of genes going up and down at different stages. And there's actually genes that coincide with the induction of critical plasticity. But which one is precisely involved in the induction of this timing is not known.
In other regions of the brain, how do you control it?
Well, we don't control it. This is just a finding that you know, you can induce plasticity when the cells acquire this age. It's an observation that probably results from the maturation of the cells. Now, we still don't understand how this happens, but if, let's say, you want to induce plasticity at certain age of the animal, what you have to do is transplant cells 33 to 35 days before that period when you want to induce the plasticity. Now, this is in mouse that have a short period of time. In humans, this is probably much more protracted, and it is known that interneurons mature very, very slowly in humans, especially some subtypes of interneurons.
Have you tried to somehow precondition the cells?
This
period of 33 to 40 days?
No. This all is very recent. This was published 2 years ago, and we are gearing up to do some of these experiments. It would be fantastic to be able to precondition them. The other very interesting question is that these cells are migrating all over the cerebral cortex, and different parts of the cerebral cortex are entering plasticity at different times, raising the question of what local factors induce the acceleration of the maturation of these cells, or whether there's unique subsets of interneurons that go to specific locations that have a different program, different timer. And that is, again, not known. Yeah. These are very exciting questions, precisely what we would love to understand.
Another exciting question? Well, Mr. Alvarez, I didn't quite understand how they move from the
How they move?
Yeah, I mean the mechanism, do they go with blood vessels or is it not, no?
So some things are known. The story is not totally clear. So the cells first move along the walls of the lateral ventricle anteriorly, and then they join the migration to the olfactory bulb, and they move, as that movie show, in chains. So they actually hop along each other. So they use each other. In English, it's called frog, like when kids jump another and they do like that. So the cells are using each other to move. There is no radial glia guides that might guide this migration. Now, there's something in there that can reform the chains, because you can remove the chains and then the pathways reform and the cells still go to the olfactory bulb. And there's two factors we have discovered in bulb. One of them is SLEET1 and SLEET2. The other one is SLEET2. And this happened in the most surprising manner, the way this works. So if this is the lateral ventricle, it is all covered by ependyma in those pinwheels that I showed you. And what this student in the lab found is that the cerebrospinal fluid moves in the same direction as the cells move forward. And it is because of the orientation of the ependymal cells. So if you disorient the ependymal cells by using mutants in which the cilia do not form normally, then the cerebrospinal fluid does not flow normally. But what was very surprising is that then the cells get disoriented. Instead of going forward, they start going ventrally, and much fewer cells go to the olfactory bulb. So that led us to suspect that the choroid plexus that is back here is a structure looks like it's epithelia that's inside the ventricles and is very involved in secreting cerebrospinal fluid. Perhaps was secreting something that was making these cells go forward towards the olfactory bulb. So what we did is take out a piece of choroid plexus and grafted it here. And now these cells began going backwards. And much fewer cells went to the olfactory bulb. And then we found that one of the factors that were secreted here was SLEAD2. So now if we put a SLEAD2, actually we can make these cells go backwards. And also what we found is that SLEAD2 is becoming a gradient, where it's very high in the back, very low in the front. So what is interesting here, and I give this to students at UCSF, is that many times you think about the factors explaining the mechanism. But in this case, it's not that. It's really the epithelial polarization that's key to the mechanism. If this epithelium is not polarized correctly, then the cells do not move forward correctly. So anyone that's interested in this was published by a postdoc in the lab. His name is Sawamoto. And it's like in the middle of the 90s, 96 we published this. And it actually describes how this polarization of this epithelium is key for the cells going forward. Now, once they're here, there is some evidence that there's chemoattractants here that keep pulling these cells going to the olfactory bulb. But this is another fascinating question that, a little bit is known, not everything is known. So now Sawamoto in his own lab has also found that SLEAD1 is actually produced by the neuroblasts themselves, and it's also chemorepellent. But what it's doing is keeping the fibroblast away, outside of the way, so that the cells can move effectively towards the olfactory bulb. If you remove SLEAD1, the migratory streams get invaded by fibroblast processes, and then the cells get impeded in their migration. So there's multiple mechanisms at work here, and this is a little bit of what's known about how these cells go to the olfactory bulb.
Do they cross the epidermis?
What?
How do they cross the epidermis?
So they don't cross the ependyma ever, because this is the cerebrospinal fluid here. Here's the ependyma with the cilia, and the cells are migrating here, underneath the ependyma. So a slit is here, and actually gets transported deep into where the cells are migrating, and is higher in the back and lower in the front. Yes?
In what age of mice's neural stem cells are active mostly, and if there is a certain age, does it mean that cortex grows more active in that period?
So you're talking about the adult stem cells?
No, in general. Maybe when they are little mice.
In development.
Yes.
So that many people, laboratories have studied, is nothing that I have studied, but during development, during embryonic, day 11 to 17 more or less, is when in the mouse most of cortical cells are made. And they are produced in a sequence, the deep first, followed by the more superficial ones. This is work from Sue McConnell. So early progenitors produce deep pyramidal cells, late progenitors produce superficial granular cells. And this is for the pallium, for the cortex. The interneurons, as I said before, actually come from the medial ganglionic eminence and migrate tangentially and actually invade the whole cortex.